Welcome Junior Tuckies, my name is Mrs. Ramlal. This video I've prepared for the grade 12s to help them with their final exam preparation. In this video we'll be looking at the November 2020 paper 2. Before you watch the video, make sure you have the question paper and memo at hand. Right. 1.2. Give the correct biological term for each of the following descriptions. Write only the term next to the question number in the answer book. Right, 121. Similar structures in different organisms indicating descent with modification. Right, your key words is similar structures, different organisms, descent with modification. So, yeah, similar structures in different organisms that indicates descent with modification. This answer can only be homologous structures. Make sure you spell this word correctly. 1.2. Large, pointed teeth in African apes used for tearing food. Now, the teeth in African apes used to tear food, that we know, is obviously canines. Right, 1, 2, 3. The part of the skull that houses the brain, that we know, cranium. 1 to 4. Non-sex chromosomes in humans, we know that as autosomes. 1 to 5. The network of genetic material found in the nucleus during interphase, that is, of course, the chromatin network. 1 to 6. The number, shape, and arrangement of all the chromosomes in the nucleus of a somatic cell, that is a karyotype. And the last one, 1 to 7, having a protruding jaw. When the jaw is pushed out or protruding, that is known as prognathous. Make sure you know how to spell prognathous correctly. Right, 1.3. Indicate whether each of the statements in column 1 apply to A only, B only, both A and B are none of the items in column 2. Write A only, B only, both A and B are none next to the question number. Right, 1.3. Long and narrow pelvis. Now we know a long and narrow pelvis is in African apes and humans have a short, broad, bowl-shaped pelvis. So our answer is obviously A only. 132. The point of attachment of two overlapping chromatids, that we know is the chiasma, and the locus is not a point of attachment. So our answer is B only. And then 1.3.3. Variation in human height. That is a very good example of continuous variation. So our answer is, of course, A only. Right. 1.4. The diagram below represents all the chromosomes in a cell that is undergoing normal cell division. 141. Name the type of cell division that is occurring in the cell in the diagram. Now, can you see in the diagram, all these chromosomes are touching at a certain place, and we know crossing over is going to take place. So what type of cell division this is? It can only be meiosis. Then, name the B phase of cell division during which the chromosomes behave as shown in the diagram. Now this is crossing over. In which phase does crossing over take place? Prophase 1. And make sure you have the number 1. 142. Where in the human female body would the type of cell division, which is meiosis, take place? So where will meiosis take place in the human body? We know the ovary. 143. Give the letter and the name of the structure that attaches to the spindle fibers. Now in the picture we know that it's the centromere, which is C, that attaches to the spindle fibers. And then 144. How many chromosomes will be found in each daughter cell at the end of the cell division? Now can you see in the picture there's 6 chromosomes. At the end in each daughter cell, there will obviously be 3 chromosomes because that's what meiosis does. It reduces the chromosome number. Right. 1.5. There is variation in the characteristics of fur color and fur texture in cats. The table below shows the alleles that control these two characteristics. So here we have the characteristic fur color, allele, phenotype. Fur color, big B, is black. Fur color, small b, white. Fur texture, big R, rough. Fur texture, small r, smooth. The Punnett square below shows the inheritance of these alleles in a genetic cross. So there's the possible gametes from the male on the top, as you can see here, and here's the possible gametes from the female. Now, 151. Name the dominant phenotype for fur color. So let's look at fur color. And what's the dominant? Of course, it is black fur, as you can see in the table here. Name the recessive phenotype for fur texture, which is the small r, and what's the phenotype? Smooth texture. Right. Then 152, give the genotype of offspring X. And that's easy. You take small b, big R, and that's how you work out the offspring. And there's your answer there as well. Then give the B phenotype of the female parent. Now the phenotype of the female parent, she is small b, big R. So she is white fur color and what texture? And rough texture. Okay. Then 152C, give the genotype of the male parent. Right. Of the male parent, can you see? Each of the gametes have a big B, so it'll be big B first. Each of the gametes have a small b as well. And then each of the gametes have a big R and a small r. So yes, you can see heterozygous is the male. And then the last one, state the phenotype of the phenotype that all the offspring of this genetic cross have in common. So can you see? All of these offspring have a big R in them. So what do they have in common? The rough texture. Right. 1.6. Scientists compare the number of differences in the amino acid sequence to see how closely related species are. Fewer differences in the amino acid sequence mean the species are more closely related. Cytochrome C is a protein that occurs in many species. The amino acid sequence of this protein differs between species. The table below shows the number of differences in the amino acid sequence of 3 species A, B, and C. Right. 161. What type of evidence for evolution is being used in this table? So here we're looking at the sequence of the proteins, the sequence of the amino acids. So that, of course, is the genetic evidence. Because remember, amino acids and proteins, that is what makes up genes. Okay. Then 162. Give the letter of the species A, B, and C that should appear in positions 1, 2, and 3. As shown in the diagram below. Now, how I will work this out? If you look at the table, species A and B have 11 differences. Okay, and the fewer the differences means they're more closely related. So obviously A and B far apart. But look at species A and C. They have only 3 differences. So I would immediately put A and C here because they have the least differences. So they are more related to each other. And then what leaves us with? Of course, B, that has the most differences between both of them. So that's how I would work it out. And you can see the memo has that option for either or. And the reason why they have 2 options is because the numbers are 11 and 10, so it's easy to be interchangeable, the answer. It could be ACB or CAB. Right. Detectives were investigating a crime scene and found blood on a broken window. They suspected that the blood was that of the criminal. To identify the criminal, they analyzed a DNA sample from the blood and compared it to that of 4 suspects. The diagram below was produced. So there you see a DNA sample, right? You see Bato, Dave, Jenny, and Tabi's DNA profile. 2-on-1. Name the technique that was used to identify the criminal. Now we know these bands are DNA bands, so this process is DNA profiling. Right. 2-on-2. Who is the possible criminal? Now what you do is you look at the DNA sample, right, that was found on the blood, and you match how many bars are in common with the DNA sample. Now, if you look, just looking at it, there's 1 bar, 2, 3, 4, 5, 6, 7, 8, 9. I can see that Jenny has the most black bands in common with the DNA sample. So Jenny is obviously the possible criminal. 2-on-3. So if you look at it, Jenny's DNA profile of bands matches the DNA profile of bands from the sample found at the crime scene. 2-on-4. State one other use of the technique identified in question 2.1.1. So they're asking one other use. So, proof of paternity, tracing missing persons, identification of genetic disorders, establishing family relations, matching tissue for organ transplants, and identifying dead persons or animals. Right. 2.2. A farmer decided to have his best meat producing bull cloned. The following steps were used in the process. A muscle cell was taken from the bull and the nucleus was removed. An ovum was taken from a cow and the nucleus was removed and discarded. The nucleus from the muscle cell was placed in the empty ovum. The ovum was given an electric shock to stimulate normal cell division to produce an embryo, and the embryo was placed in the uterus of a surrogate cow where it developed into a clone. 2.2.1. What is cloning? So cloning is the production of a genetically identical organism, as you can see here in the memo. 2.2.2. Why the nucleus of a muscle cell was used and not the nucleus of a sperm cell? Well, that's easy. It's the muscle cell that contains the genetic material, and it's diploid. And the sperm cell only has half of the genetic material, right? Or is haploid. Okay. 2.2.3. Explain why the nucleus of the ovum was removed. Right. To remove the genetic material of the cow, right, so that only the genetic material from the best meat-producing bull is present. Of course, what happened? They removed the nucleus of the ovum. So you want to make sure that only the desirable trait is present. So that is why you remove the nucleus of the ovum. Another reason could have been you don't want the DNA from the nucleus to affect the outcome as well. Okay. 2.2.4. State one benefit of cloning. To produce organisms with desired traits. Example when it comes to health, appearance, nutrition, yield, shelf life, conservation of threatened species, and to create tissues or organs for transplant. Right. 2.3. A man with blood group AB and a woman who is heterozygous for blood group B plan to have children. 2.3.1. How many alleles control the inheritance of blood groups? That's easy. 3. 2.3.2. Describe the type of dominance that occurs in the inheritance of blood group B in the woman. So, a blood group B is definitely complete dominance. Why? The allele for blood group B is dominant over the allele of blood group O because blood group O is obviously recessive. And then 2.3.3. Use a genetic cross to show all the possible genotypes and phenotypes of their children. So, as you see in the memo, this is how you set it up. P1 phenotype, blood group AB, and blood group B. That's the parents. Then you have genotype, IA, IB, IB, and small I. As you can see in the memo, meiosis, gametes, fertilization. You can do your answer like this in the memo, or you can use a Punnett square. I prefer to use a Punnett square. So can you see the gametes here? It will be one child with IAB, IAIB, IBIB, IA small I, IB small I. So you got marks here for P1, F1, meiosis, and fertilization. And you must show all the correct gametes and genotypes. Nice question. All right. 2.4. Sickle cell disease is caused by a recessive allele and first appeared in humans as a result of a gene mutation. The table below shows the number of children born with sickle cell disease in some regions in a particular year. So there you have the region, the Democratic Republic of Congo, United States of America, Nigeria, United Kingdom, Tanzania, other, and the worldwide total. Then the number of children born with sickle cell disease. 2.4.1. What is a gene mutation? A gene mutation is a change in the sequence of the nitrogenous base or nucleotides of a gene. 2.4.2. Which region has the highest number of children born with sickle cell disease in that year? Now, if you look at your numbers, obviously, can you see? It is obviously Nigeria. 2.4.3. What percentage of the worldwide total of children born with sickle cell disease came from the Democratic Republic of Congo? Show all calculations. So, the DRC, it will be 397846 over the total multiplied by 100, and your answer is 13%. 2.4.4. Use the letters big D and small d to give the genotype of a person who suffers from sickle cell disease. So it's a recessive allele, right? So small d will be sickle cell disease. And then use the letters big D, small d to give the genotype of a person who carries the allele but does not suffer from the disease. Of course, that person must be heterozygous, so big D, small d. Right. 2.5. Gault syndrome is a sex-linked genetic disorder. It's caused by a dominant allele, X big G. The diagram below shows the inheritance of Gault syndrome in a family. 251. Name the type of diagram shown here. So here you can see the inheritance of a characteristic over generations. So this is a pedigree diagram. Right. 252. How many females are in this family? Now the females are the ones that are actually round. So 1, 2, 3, 4, 5, 6. And 252b, how many males in the F1 generation have Gault syndrome? So if you look in the F1 generation, so F1 generation is this. How many males have Gault syndrome? 1. Right. 253, give Gabby's genotype. Now, Gabby is affected. So she would definitely be X big G. But can you see Gabby has 2 children that are not affected. So she must carry the normal gene as well. So she is heterozygous. Right. 254, Anju and Pelusa have 4 children. Give the phenotype of their sons. So I'm just going to do the Punnett square. So X big G Y is Pelusa. Right. X small g is Anju because she's normal and she's a female. And Pelusa is a male, so is X big G and is affected. Remember, this condition is dominant. So they want to know the phenotype of their sons. So X small g y, X small g y is their sons. So both their sons are obviously normal because small g is normal. Right. Then 255, they say explain your answer. So we say it. Pelusa is affected, has X big G Y. Anju is unaffected and so has X small g small g. Males, can you see, inherit the Y chromosome from the Pelusa, which is the father, and they inherit the X small g from Anju, hence they're normal. So you can do the cross to help write your answers, but they said explain. They didn't say use a Punnett square. So make sure you still write it out. 3.1. Describe the process of natural selection. Very easy, straightforward, nothing to apply, so you just have to regurgitate what is in the exam guidelines. Organisms produce a large number of offspring. There's variation amongst the offspring. Some organisms have favorable characteristics and some do not. When there's a change in the environmental condition, there's competition. All right? Or when there's competition, organisms with favorable characteristics will survive, while the organisms with unfavorable characteristics will die. The organisms that survive will reproduce and pass on the allele to the favorable characteristic to their offspring, and then the next generation will therefore have a higher proportion of individuals with a favorable characteristic. Any 7 you could have had. All right. 3.2. Fossil evidence for many years may be interpreted in different ways. One possible model of human evolution is shown below. 321. Name the family to which all of the represented organisms belong. So these are all the different organisms that you see, all the ancestors of humans, right? And so which family they belong to? Hominidae. Okay, right, and make sure you spell it correctly. 322. Describe how cultural evidence is used to support the theory of human evolution. 2 marks. Cultural evidence is evidence such as tools, weapons, language, artifacts, and it's used to show advances in human development, especially in the advances in the changes in the size of the brain. 323. How long did the most recent common ancestor of Homo erectus and Homo heidelbergensis exist on Earth? So let's read the question again. How long ago did the most recent common ancestor of Homo erectus and Homo heidelbergensis exist on Earth. So let's look for Homo erectus. Here it is here. Heidelberg Genesis here. Now let's look for their common ancestor. So we look at where the two lines meet. So there's it here. And guess what? Your answer is 3 MYA, which is million years ago. Right. 324. Explain a possible reason why Homo ergaster was placed between Afarensis and Heidelberg Genesis on the model. So it was placed between Afarensis and Heidelberg Genesis in the model. So it probably Homo ergaster shows characteristics of both Australopithecus afarensis and Heidelberg Genesis. So it's possible that it is a transitional species. Right. And then 325. Explain how the fossils of organisms that exist from 4 million years ago to present time are used to support the out of Africa hypothesis. And this is also in the exam guidelines, so you must make sure you memorize this. So, the fossils of Australopithecus were only found in Africa. The fossils of Homo habilis also were found in Africa. The oldest fossils of Homo erectus are found in Africa, and the oldest fossils of Homo sapien are found in Africa. This suggests that the ancestors of Homo sapiens originated in Africa. Can you see the word only? You make sure you have the words only. Right, 3.3. Many long-tailed widow birds have extremely long tail feathers that they use in mating displays to attract females. Scientists conducted an investigation to determine the relationship between the length of the male long-tailed widow bird's tail and its mating success. The procedure was as follows. A total of 27 male long-tailed widow birds were sampled and divided into 3 equal groups. The tail feathers of the birds in each group were treated in the following way. Group 1 was cut short. Group 2 made longer by adding artificial feathers, and Group 3 left unchanged. The 3 groups of male long-tailed widow birds along with female long-tailed widow birds were released into environments suitable for mating. Each time a pair mated successfully, they produced a nest and all the nests were counted. The average number of nests produced by each group were calculated and used as an indication of mating success. The results are shown in the table below. Group 1, 2 and 3 and the average number of nests. So 331. Name the reproductive isolating mechanism that occurs in long-tailed widow birds. So of course, right, it's species-specific courtship behavior. Right, so it's a behavior used to attract females that's only specific to a species. Then name the independent variable in this investigation. What did you choose to control? You chose to control the length of the male long-tailed widow bird's tail. It's very weird with all the tail. So length of the male long-tailed widow bird's tails. And that obviously is in brackets, as you will see. Write it in the memo, or you can leave it as is. Right, 332. Explain why 27 long-tailed widow birds were used in the investigation instead of only 3. Now, we already know it's going to be a larger sample size, which will increase the reliability of the investigation. 3.3.3. Explain why Group 3 was included in the investigation. This we already know. This Group 3 serves as a control, so it can be compared with the other groups and show that the tail length is the only factor that affects the results, or you could have said improves the validity of the investigation. 334. Draw a bar graph to represent the results of this investigation. So there you have your bar graph with the average number of nests produced, your group, right? Your bars are the same width, the spaces are the same, the average number of nests produced by each group of birds, and then you got a mark for the correct bar graph drawn. Caption was correct, axes X and Y were correctly labeled, the scale was correct, the bars were the same space and the width, and all 3 bars were plotted successfully or correctly. And 335, state a conclusion for this investigation. Now, you could have said the longer the male long-tailed widow bird's tail, the higher the mating success, or the other way around. The shorter the male long-tailed widow bird's tail is, the lower the mating success, as you can see. Okay, right. Pottos and lemurs are small mammals. Scientists believe that the pottos and lemurs share a common ancestor that existed in Africa. Presently, pottos only occur in Africa, while lemurs are only found in Madagascar. Madagascar is an island off the east coast of Africa, as shown in the diagram below. 341. Explain how continental drift could have affected the distribution of the common ancestor. So, there was once one large continent, and the common ancestor existed throughout the continent. When Madagascar separated, the common ancestor then was found in both regions. 342. Describe the speciation of the pottos and lemurs to become different species. The common ancestor became separated into two groups by the ocean. There was no gene flow between the two groups. Each group experienced different environmental conditions and underwent natural selection independently. The individuals in each group became different. How? Genotypically and phenotypically different to form the pottos and lemurs. Eventually, if the two groups are mixed again, they cannot interbreed as they are different species. Right, and then the essay question. I still do the essay question because this can still be asked. Each part can be broken down and given as a short question. Question 4. Describe the location and structure of the DNA, the process of DNA replication, and the significance of this process for mitosis. So, where is the location of the DNA? The location of the DNA is in the nucleus, mitochondria, and chloroplast. The structure of the DNA is double-stranded. It forms a helix. It's made up of nucleotides. Each nucleotide has a deoxyribose sugar, a phosphate group, and a nitrogenous base. The bases are adenine, thymine, cytosine, and guanine, which join to form complementary bases, or you could have said adenine pairs to thymine and cytosine to guanine, held by hydrogen bonds. DNA replication. The DNA double helix unwinds and unzips, or the weak hydrogen bonds break to form two separate strands. Both DNA strands serve as templates to build a complementary DNA. Using free DNA nucleotides from the nucleoplasm, this results in two genetically identical DNA molecules. Each molecule consists of one original strand and one new strand. What is the significance of DNA replication for mitosis? The genetic material or DNA is doubled so that each cell receives the same amount of DNA to ensure that all the daughter cells are genetically identical. That was a nice question. Right. Thank you, grade 12, for watching this video. I hope you learned a lot. Join me and watch all my other videos as well.